I'm Yu Junlin, in case you are in the right classroom, okay? So I'm one of the 6 faculty members who are in charge of advanced cell biology course. So today I will introduce 2 chapters, Chapter 10, Biomembrane Structure and the Transmembrane Transport of Ion and the Small Molecules. And that would be the Chapter 11. And next we will talk about the Chapter 20, Integrating Cells into Tissues. After my part, the Dr. Liao will give 2 more lectures about the Chapter 13 and Chapter 16. And it will come with the first exam to cover these 4 courses, okay? Yeah, just keep in mind. So we don't ask you to buy the textbook, but the content is mainly based on these 3 textbooks: Molecular Cell Biology by Harvey Lodish, in case you want to read more, okay? Or Molecular Biology of the Cell by Bruce Albers, or Cell Biology by Jurek Karp, okay? As you can see here, my slide is pretty busy, but it just kind of help you to know the key concept of the content of this slide. For example, those red sentence, they will kind of write down the key concept or description about the slide. And the blue one will point out or highlight the key point about the figure. So hope it can help you to remember what's the important part in this slide, okay, after class. If you have any question during the class, just raise your hand and let me know. So because this is a cell biology course, so begin this course, before we go into the detail of this course, I would like to kind of discuss about this question: why we need to study cell biology. Okay, why cell biology is so important? Now we are in the College of Life Science and Medicine, right? So what is the fundamental question in life science biology or medicine? Okay, to me, because in this college, many people use different model organisms. Some faculty, they use zebrafish, Drosophila, or mice, or plant, or virus, or C. elegans. They study different topics: immunology, neuroscience, evolution, blah, 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 a lot of different things. But the main question people always care about is, to me, this is my answer: what is life? How does life work? Actually, we don't have a correct answer yet, okay? But as a cell biologist, I probably have my answer. Why cell biology is so important? Because if you check this, please check this figure, you will see the figure just showed how the simple atoms, step by step, and finally formed, kind of assembled to build up the whole organism. And to me, there's a barrier here. Beyond these barriers, everything are dead stuff. It's dead stuff, okay? But after these barriers, everything are living things, okay? So on the other word, the cell is the basic unit of the life. Moreover, everything inside the cell is dead stuff. If you want to control the dead stuff, you need to follow the universal principle. You need to follow the physical or chemical principle. For example, if I want to hold this water bottle, I need to use my hand and follow the universal principle, right? It's also the case for the cell. Cell need to control everything, dead stuff inside the cell, and conduct the life, kind of generate the life environment and conduct such a living things. So they need to follow the physical or chemical principle to control everything and build up the kind of living environment. So it's all about pH level, it's all about the charge, it's all about the hydrophobicity, affinity interaction, something like this, okay? But finally, using such a simple universal principle, they can build a super complicated living environment and activity. That's amazing. And that is what we want to learn the cell biology. So let me show you the videos. So this is kind of called the MIT chair. So the researcher from the MIT, they just put the several blocks. So the end of those blocks, they have affinity or can bind to the other end of the block. Of course, their binding is kind of pairing, and it depends on their design. And they just stir the water to kind of randomly mix this block. After 7 hours, finally they can build up such a chair. They call this MIT chair. Okay, this chair is important because it's from MIT. but it's just simple video or experiment to tell you through the random mixture or just this environment, if they have a kind of designed interaction between the different block or elements, they can build up what they want. It's also inside the cell. Actually, a cell can do the same kind of process and even crazier or more complicated. For example, the ribosome, they can synthesize this protein and also other protein. And this protein, through the chaperone protein binding, they will change their conformation, and then they will become have affinity with other chaperone, and then they will have kind of keep changing their conformation step by step, and finally they have affinity to bind with other protein. So through this, cell can generate functional protein complex inside the cell. But for the experiment I showed you before, it's only put 5 or 6 blocks, right? But inside the cell, in the cytosol, over 1,000 protein mixed together and keep doing this, okay? And assemble this protein complex usually take a couple seconds and minutes. That's amazing. Okay, so I just encourage you, when you start the cell biology, just keep in mind, always think about how cell conduct such a different activity based on universal principle. Okay, that would be my mindset. Okay, so let us talk about the chapter 10, biomembrane structure. So again, as I mentioned before, cell is the fundamental unit, basic unit of the life. Everything here happens is living environment. So to conduct the living activity, cell need to do the metabolism, cell need to do this signaling. To achieve that, they need to uptake nutrient or essential molecule to conduct those activity. But after those reaction, of course, they need to release the waste or exchange molecules. Okay, so all this molecule exchanging happens in this plasma membrane or the boundary to separate the cell and extracellular space. Okay, let me enlarge this area. This is the side view of the plasma membrane. Okay, basically, based on the physically or chemically, you can consider plasma membrane is a hydrophobic core. It's just a layer with a hydrophobic core. Okay, it's all about the chapter 10, chapter 11. This is a key concept. This is a hydrophobic core. So after this course, you should know how do molecules transport within or across the membrane, and how do protein locate on the membrane stably. They all need to deal with this hydrophobic core. Okay, that will be the key concept. We will keep remind you and point it out. Okay, so the first, they just showed you the overview of the plasma membrane, what kind of component you can find from the plasma membrane. First, the cell membrane is composed of many different lipids and proteins. Okay, so the majority of the lipids are shown by the gray color here are phospholipid. It's a lipid conjugate contain the phosphate group. Okay, so we call phospholipid. It depends on the way you can see the different protein they will incorporate into the membrane through the different strategy. So it depends on the way they incorporate into the membrane. So the protein can be classified into the 3 groups: periphery membrane proteins, and the integrate membrane protein, or we call the transmembrane protein, and the lipid anchor protein. We will discuss more detail in next about the protein in next chapter. Okay, but here we will focus on the phospholipid first, and about how this is the cartoon we all learned from the textbook. But maybe 100 years ago, people really don't know what the cell membrane is. Okay, and what's the physical or chemical property of those lipids even. Okay, so about in the 1880s, several physics, they just try to do the experiment to characterize what's the property of the lipid. And they just put the many lipids to the water tank, and they found that those lipids were stayed on the surface of the water like this. This is the side view of that water tank. Okay, and also the polar head of the lipid will just bind to the surface of the water. Okay, and there's a barrier. Okay, So they can kind of use this barrier to concentrate the lipid in that water tank only on the surface, or just spread out, okay? So they found, based on this experiment, they found that those lipid, they will use the hydrophilic part to align with other hydrophilic part. Hydrophobic part align with other hydrophobic part. And finally, they will align with each other with the same orientation. That will be the really basic information about the lipid bilayer, okay? And they gradually know the physical property of the lipid, okay? And after about 50 years later, based on the electron microscopy study, they found many different cells from different species. They all have the same cell membrane, okay? This structure here, here, and here, okay? And then people just do a lot of experiments, and finally they try to describe, because those stuff are pretty tiny, it's way below kind of resolution of the light or light microscopy. It only can be observed by the electron microscopy, but those cells can be only observed by electron microscopy after fixation. So they are dead. You don't see their dynamic, okay? You only know their structure, but you don't know the dynamic. But based on several different experiments, people just propose the model to describe how this lipid, how this protein, membrane protein, behave on the cell membrane. So that will be more familiar with you. The Singer and the Nicholson in 1972, they proposed a model called the fluid mosaic models. So this is the video kind of describe the fluid mosaic models. First, what is the fluid? Okay, so they describe the lipid and the protein. They can behave in the kind of like a freely diffuse in this two-dimensional fluid. This is a side view of those two-dimensional fluid. Okay, they can freely diffuse here. They cannot go to the 3D. They only can diffuse in this two-dimensional sheet. Okay, and what's the mosaic come from? So this is not the kind of even distribute. Okay, some protein or some lipid they can concentrate or form the cluster, like here. Okay, there will be higher concentrate and more protein here. So when you observe, you can kind of imagine that when you observe those protein or lipid pattern, it kind of form the mosaic pattern, and they are diffuse. Okay, that is why it's called the fluid mosaic model to describe this. Okay, then the textbook just give you more idea how the membrane behave, so or the property. So the first, the eukaryotic membrane can be really dynamic. They are not static. They are super dynamic. Okay. For example, and then the structure and the shape can bend continuously. For example, this is the surface of a cell which has been infected by HIV virus. So HIV virus will infect the mammalian cell and kind of use their genome to generate their component. And after a while, they will assemble together, and finally they will accumulate to the surface of that mammalian cell and try to build up the more HIV virus. And they use the plasma membrane, which is the cell membrane, okay, as their kind of as their core, okay, just budding out to form the new virus. Okay, so this is just use EN to show you the beautiful shape of the cell membrane here. And also this is the Golgi apparatus, which is an important organelle here. You can also see the cisternae organelle, cisternae structure here is all covered by the membrane structure. So the membrane structure, it can change a lot, okay, to the different shape. And also it's super dynamic. When you check, this is the kind of top view of the live cells. As you can see here, this is a migrated cell. And they keep doing kind of, you can see those are red signal, they are membrane, they're label membrane. They keep moving and to do such a wave-like behavior. We call this membrane ruffling. And such a membrane ruffling is kind of, you need to keep changing the membrane component, and that will be the important way for cell to migrate toward their destination. Okay, they are not like this. They keep changing their membrane. Okay, so you will see the whole membrane like this. They are super dynamic. They are not static. And also, the membrane can change their shape to many different ways. For example, the red blood cells, you will see those are disc-shaped structure here is all covered by the membrane. This is one red blood cell. And if you zoom in several cells, you will see kind of some surface of the cell, they will have this hair-like structure we call the multicilia, especially in our airway. They will kind of multicilia will keep moving and help you to kind of remove the mucus, okay? And this is the flagella. In the sperm, you will see they also kind of, you will see they kind of appear as a really different shape, right? They have a super long tail. And also, this will be the other extension of the cell is primicilia. It's happened, you can find this primicilia in over 90% of the cell in your body, but one cell only have one primicilia. Okay, they are kind of function as a cell antenna to receive the extracellular signaling. It could be the mechanical signaling, it could be the chemical signaling. Because why this tiny hair, only one antenna is so important? People believe, or I believe, there are many different signaling receptors concentrate in this tiny area. So they will be, when the ligand or cue come here, they will be easier for them to do the kind of crosstalk among the different signaling pathway. That is why the primicilia is important. Because people may ask, there are a lot of several signaling receptors here, but they could also localize to the plasma membrane, right? But in the plasma membrane, the local concentration or local amount of those receptors is low. So they don't meet with other signaling pathway or receptor easily. So it's not easy for them to do the crosstalking. But in the primicilia, there will be the spatial place for them to do the crosstalking and finally trigger the downstream really complicated signaling, okay? And all this extension, even they kind of stick out really kind of with a really long shape, they all covered by the membrane, okay? So the membrane could be really dynamic and really kind of versatile to change their shape. Okay, so the major component, as I mentioned before, is phospholipid. And the phospholipid is the ketone of the phospholipid. And we here call the phospholipid are amphipathic. What is amphipathic? Amphipathic means molecule contain two parts with very different chemical property. Here would be the hydrophobic and hydrophilic. Okay, so in this is ketone and this is the real chemical structure of the phospholipid. The polar head will be, we call here hydrophilic head or polar head. It's super hydrophilic. And this will be the two hydrophobic tail, which is super hydrophobic. So you have a right kind of opposite chemical property, hydrophobic and hydrophilic in the same component. So we call this amphipathic molecules. Okay. So as I mentioned before, people already know through kind of the experiment over this century, hydrophilic molecule align with other hydrophilic, hydrophobic align with other hydrophobic. So if you put those lipid kind of in the aqueous solution, outside the cell, it's a water solution, it's an aqueous solution. In the cytosol, it's also aqueous solution. So the way they kind of will build up kind of energetic favorable condition will be like this. They face their polar head, which is the hydrophilic part, to the hydrophilic extracellular space or the cytosol part. And they align their hydrophobic tail together and form this lipid bilayer. So we call this one leaf lap and the other leaf lap. And the thickness of this leaf lap can be measured by the electron microscopy. So you can use osmi, OSO4, which is a heavy metal, which will strongly bind to those polar head here. So you see there's one line and the other line, which just bind here. Okay, and then based on the electron microscopy, you have a good enough resolution to observe this two leaf lap, which is about 3 to 4 nanometers. Okay, this will be the thickness of the lipid bilayer. So this just ketone to really quickly show you how they form the cell or the different lipid bilayer structure. For example, if you mix those lipids together, they will gradually align with each other based on the hydrophilic or hydrophobic. So it will have the red color here will be the hydrophilic polar head layer. The white one will be the hydrophobic layer, hydrophobic core. And the other side, of course, will be the also polar hydrophilic polar head. But when you put to the aqueous or water solution, they don't really like this because the hydrophobic layer were exposed to the water, right? So this is not an energetic favorable condition. So they will try to hide those hydrophobic core, and finally they become a bowl. If the lipid concentration or lipid molecule is good enough, or they have enough those lipid molecule, they can form still can form the bowl. This is a bowl like with two leaflet. We call this liposome. If they only don't have enough kind of lipid to form the bowl, they will form the smaller bowl, which is my cell. Again, they still expose their kind of hydrophobic polar head to the outside, to the surface, and face their hydrophobic fatty acid tail in the core. Okay, they always try to hide their hydrophobic core inside. They don't expose the hydrophobic core to the solution. Okay, and the liposome will be kind of really popular. Not that popular recently. Recently, it's more popular by some lipid capsule or nanolipid, kind of nanoparticle. Okay, but liposome is pretty popular before. They can kind of, if you mix those lipid with antibody or the drug, they kind of can serve as a vehicle to deliver drug. Okay, you can put to your bodies, and it kind of will be easier to go to the target site. Okay, because you need to deal with the bloodstream. In the bloodstream is hydrophilic environments, but your drug could be sometimes maybe the hydrophobic. Okay, so by doing this, you will have a stable, and they can concentrate more drug together in the one dose. Okay, so the biomembrane contains three principal classes of the lipid. One is phosphoglyceride, and second is sphingolipid, and third is sterol. So for the phosphoglyceride, you will see this is the kind of major structure of the phosphoglyceride. They have a two hydrophobic tail, which is based on the fatty acid tail. Okay, they are super hydrophobic. And here, it will be one glycerol and conjugate with different polar head. And depends on the polar head, they will have a different name. This will be the phosphatidylethylamine, stand for, will be the P in short, or phosphatidylcholine PC, or phosphatidylserine PS, phosphatidylinositol PI. It depends on whether it has a charge or not. This PE will finally become the no charge PC. PS and the PI will be negative charge. Okay, and also there will be the other kind of, this plasmalogen is a one subtype of the phosphoglyceride. It's just one subtype. It's also belong to the phosphoglyceride, but it's kind of one subtype. But this phosphoglyceride is kind of special because it's usually the other phosphoglyceride, they use ester linkage. Okay, and this plasmalogen, they use ether linkage. It's more stable through this linkage. This is not so stable compared to the ether linkage. So plasmalogen is kind of, as I mentioned before, it's a subtype of the phosphoglyceride, and it's more stable. So usually you can find them their major job is kind of provide mechanical support of the membrane. You can find it in the neuron cell or heart. They just usually provide a physical stability. Okay, and it's not easy for cell to form such an ether linkage because they also need other organelle like a peroxisome. We probably can, if you are interested, we can go through more detail, but I probably will skip that part. But usually the ER can form the phosphoglyceride and use this ester linkage, but it's not so stable. We will introduce one more. Why we call that not stable, not or stable? Because there will be the one enzyme can help to disrupt or damage those phosphoglyceride. We will introduce today, okay? It's called phospholipids. So those are lipids. Why they need to kind of disrupt or cut off this phosphoglyceride? Because sometimes those phosphoglyceride may damage. They need to do kind of recycle. They need to do the metabolism. So you need to kind of really cut off the damaged one, and they can kind of for the reuse. Okay, and ester linkage will be easy kind of cleavage by the phospholipids, but the ester linkage will not. Okay, so that is why we call this more stable than ester linkage. And the sphingolipid, they share the pretty similar structure, as you can see here, compared to phosphoglyceride. They also have a 2 long tail. But for the phosphoglyceride, they use a fatty acid tail. But here they use a sphingosine as their 2 hydrophobic tail. Okay? And so that is why all sphingolipid is come from sphingosine. This will be their 2 tail. And sphingomyelin is the most abundant sphingolipid, which is here. Okay? And also, those sphingosine can also conjugate with certain sugar, like here. And it become, we call that glyco is sugar, glycosphingolipid. For example, this glycosylceramide, okay, it would be the one really typical glycolipid. So glycolipid contain 2 to 10% of the total lipid in plasma membrane, okay. And the third class group is the sterol. So there will be 3 kind of sterols, 3 types of sterol: cholesterol, ergosterol, and stigma sterol. Okay, so they share the pretty similar structure. We will see here they will have a 4 ring showing in the yellow here, and those are cholesterol. They are also amphipathic because here is a polar group. This polar group are hydrophilic, and this will be their non-polar hydrocarbon tail. Okay, and also this ring structure will also consider hydrophobic. So this part will be the hydrophobic, and this part will be the hydrophilic. So they are also amphipathic. But those cholesterol is abundant, but they cannot form lipid bilayer by themselves. Okay, because their kind of hydrophilic part is too small, and hydrophobic part is too big. So they cannot really form such a stable lipid bilayer. They usually will kind of integrate with other phospholipids. So this is a cholesterol. But again, they still use a hydrophilic part to align with the hydrophilic part of the other polar halide, and use this part will be the hydrophobic part to align with the fatty acid tail. Okay, so they will help, and then they can also generate several space for those phospholipids and increase for we call the membrane fluidity. So those phospholipids, they will have more space for them to move around and increase such a fluidity. Okay, and also cholesterol can be also found in the animals, but cannot to found in the plant or the eukaryotic E. coli. Okay, prokaryotic cells. Sorry. Yeah, we can only find cholesterol in the animals. So next we will talk about the membrane fluidity. Okay, so because you need to keep the membrane, as I mentioned before, the membrane is the boundary or the place for the molecule exchange, and then it's super dynamic, and also it's covered by the whole surface of the cell, no matter what's the shape of those cells. So the fluidity keep in the physiological. The membrane cannot too viscous, which means too low, fluidity is too low, or it cannot too kind of strong fluidity. Because if the fluidity is too high, the membrane cannot maintain their physical, cannot support their structure, right? But if the fluidity is too low, they cannot exchange molecule. They cannot change their shape. Okay, so they lost their function. Okay, so you need to keep them in the physiological range. So what kind of factor determine their fluidity? First is lipid composition. So those will be the unsaturated lipid. You will see the one typical characteristic of those unsaturated lipid is they will bend their fatty acid tail. Okay, so they will keep other lipid away from them, so they will generate some space and they will have a better fluidity because they have space to move around. But if your lipid always contains most of the saturated lipid, their fatty acid tail is super straight, so they really compare with each other. They don't have space to move around. So in this case, it will be super low fluidity. Okay, and also, in addition to the lipid composition, temperature will be also important factor to determine their fluidity. As you know, the lipid, if you put to the high temperature, heat will increase their fluidity. Okay, they will become solution. But if you put there to the freezer, they become kind of gel-like pattern, right? So low temperature will reduce their fluidity. But this is not good for certain kind of species to adapt to the extremely difficult environment. For example, some E. coli, they live in the hot spring, super hot, right? How can they keep this good fluidity and still survive there? Or some fish may survive in the super cold environment. How can they keep the good proper fluidity? So the cholesterol can help. So if you contain the cholesterol, it will narrow down even in the super high or super low temperature environment. They can keep their fluidity in the proper kind of range. Can help them, those animals, to survive there. Okay. Why cholesterol? Because they are not so sensitive to the temperature. The phospholipids, they are more sensitive to temperature. If you use really high or low temperatures, it will change it was really easy to damage those phospholipids or sphingolipids, and their orientation will also change. But the cholesterol is not so, they kind of can tolerance to those different temperature compared to the phospholipid. Okay? And also, the dynamic of those, people believe the lipid, they only can do, will be easier to do the lateral movement, okay, just through this direction, the movement through this direction. They cannot easily do such a vertical movement. It's called the flipping, okay? Why? Because if you want to do this, flip to the other lip flap, you need this polar head hydrophilic part need to expose to the hydrophobic part. Then they can reach to the other hydrophilic layer. It's not an energetic favorable condition. Okay, such a hydrophobic core will prevent them to do the vertical flipping. Okay, that is why they usually just do the electron diffusion without any other energy. And the speed is about 1 micrometer per second. So it depends, of course, different cells have different size, but usually the mammalian cell, the diameter is about 10 to 20 micrometers. So theoretically, if they need, one lipid molecule can transfer from one side to the other end within 30 seconds or 10 seconds. It's pretty fast, okay? So it will be easy. So you need to know such a temporal resolution or the dynamic. Then when you kind of check your experimental result, you will see the readout is fast or slow. You may have a better guess or have a sense it's through the dynamic or through the different mechanism. Because, for example, if you see the one reaction is super fast, I would say it could be your proteins result mainly regulated by the dynamic or phosphorylation or dephosphorylation. If one reaction could be really happens slowly, it could be the expression level issue. Okay, for example, you just treated one drug and the cell die, and you want to see whether it died only if it died or within 10 seconds. It's nothing about the protein expression, because protein expression regulation is too slow. When cell receive the external stimulation, they will gradually go into the DNA, they activate the DNA, right? And they do the transcription translation. Usually take more than 10 minutes. And if you want to use Western blot to detect induction expression or up-regulation of your protein or down-regulation of your protein, it could take about 2 hours. So everything happened within 2 hours. My gut feeling is not regulated through the protein expression level. It's through post-translation modification, it's through the others, phosphorylation, dephosphorylation, or it just translocate to the other side, inactive side. So it makes sense, it's logic, so you need to understand the speed and the dynamic. I think that's important. Okay, not only a positive or negative, it's significant or not significant. Temporal resolution, temporal process is also the important. Okay? So how to measure those molecular dynamic? Here we really typically use a fluorescence recovery after photo bleaching, or FRAP in short. So this, would be kind of one cell which we don't label anything, okay? So they have a membrane component, but we don't label, so we cannot visualize those components. They are transparent. So usually we use a chemical dye or use other ways to fluorescent dye to label or target a lipid. So therefore we can visualize. They will show us a fluorescent signal under microscopy. But this is cartoon. We don't usually we don't discuss about the single particle image now, but usually with conventional microscopy, we don't have a resolution to separate each single dot. So this is cartoon. We always see the whole cell is bright, okay? But how can you kind of really measure their dynamic? The way is you just shine strong laser kind of with the region you are interested, okay? So shining, there's a kind of mechanism. If you shine the strong laser to the fluorescence module, you will kind of make them dimmer. That's called the photo bleaching. But molecules still there. They just become invisible. They don't have fluorescence signal anymore. Okay, so this part you will lower their fluorescence signal, but outside this surface, because they only shine laser here, you only trigger the photo bleaching here. So outside this region of the area, they still have fluorescence signal, but they keep moving, right? So then you turn off the laser, just keep continuous monitoring their dynamic. You will see the invisible one still go out and go in, and the visible one will gradually go in and go out. You cannot see the invisible one. You only measure the visible one. So depends on how fast they can move. You will see the signal gradually accumulate back, or the signal recover back. Okay, that is what we call the fluorescent recovery after photo bleaching. So if you measure, draw the outline and measure the fluorescent intensity here. In the beginning, this is the experiment over the time. In the beginning, it's the original fluorescent signal. You shine the laser within usually couple 3 to 10 seconds will be enough. You will see it trigger the photo bleaching. Fluorescent become really weak, and then you turn off the laser and keep monitor what happened to those fluorescent signal in region of interest. You will see the recovery back. Okay, so their dynamic depends on, if you kind of calculate the scope, there will be the recovery rate. If they go really fast, they will recover super fast. If they those molecules move really slow, they will recover really, really slow. Okay, and also one more information we can get is they usually many cases they don't kind of recover back to the original level. They don't go back here. They only recover back to the 50%. Okay, we here code is a mobile fraction. It means in this whole population, only half of the molecule they are moving. The rest of the molecule they don't move. They are static. Okay, that is why you never see they recover back to the original level. Okay, so we call this a mobile fraction. It also can the other factor to show you how dynamic of your molecules. So we get the two information recovery rates and the mobile fraction based on those are fluorescent recovery after photobleaching or FRAP. So just give you the example. This is the endoplasmic reticulum ER, and this is the nucleic envelope. So Jennifer Lipid-Schwanz, they will shine laser photobleach here. Okay, just take a look. You will see the one recover back, right? The other recover really slow. That is mean which one move faster? ER, right? This one is super slow. They have a low mobile fraction. You can consider most of them could be the static. Okay, and this is the gauge apparatus. They just shine laser here. You will see they recover back. Okay, so I just want to mention, as I showed you before, the gauge apparatus, you will see the different cisternae, right? Like a cis, middle, and a trans, right? Do they connect with each other? How can they communicate with each other? Do you know? Through vesicle or through they gradually change kind of maturation model. Do you know that? Vesicle model or maturation model? The the protein need to synthesize in ER, then transfer to the Golgi, right? They will arrive the cis Golgi first, then go to the middle cisternae, then go to the trans Golgi, then go to the destination. So how can these different cisternae communicate with each other?
They have channels.
Yeah, what?
They have channels.
Channels?
Channels mean connection.
What kind of connection?
Like the channels. You know?
Oh, channel.
Yeah.
Channel is a protein or it's based on, channel is a really rough term.
They are membrane extension, something like that.
So you mean this one connect with this one?
Yeah.
Okay.
Small part. Yeah.
Okay, how about, can this one connect with this one?
No. This
It's only connect to the neighbor one.
Yeah, yes.
Okay, just half right. So they have a kind of membrane directly connect with the other cisternae. But in some cases, they can also connect with here. Yeah, because now we have a better electron microscope, they can do the series dissection, okay? And finally, the 3D kind of reconstruction, they show they have a kind of really beautiful cisternae, but somehow they have several kind of membrane tube can connect with each other. So it's not maturation, it could through the vesicle because there are always a lot of vesicle around here, right? But they actually physically connect with each other. So why I asked it, because if you photo bleach here, for example, here, once they connect with each other, it will always cover back to the whole Golgi. It will not always, for example, if this is the middle cisternae, you just bleach here, the molecule from the trans or cis, they don't go back to the middle, okay? Yeah. Any question? Okay. Okay, so the cell membrane is asymmetric. What is asymmetry means? Asymmetry means they have a different kind of composition, okay? For example, you will see this cartoon, they will have two colors for their leaf left. The one labeled in the orange, the other one labeled in the gray. The orange one is always face, you can see here on the surface of the lumen or in the cytosol side of the plasma membrane, they always face to the cytosol. We call that cytosolic face or cytosolic leaf left. And the other side will be the exoplasmic leaf left. And it will be the surface of the plasma membrane or also the lumen of the organelle. Okay? They have a different lipid composition. So such a kind of orientation kind of determine when lipids synthesize in the ER. ER is the major site to synthesize lipids. Okay? When you kind of read this sentence, certain reaction only happens to certain organelle, or such a reaction mainly happens to that organelle, which means there will be the key enzyme, key player only exists in the ER. Okay, so there will be the several key enzymes only exist in the ER. That is why ER will be the major organelle to synthesize lipid. You only can find those lipid synthesized protein in the ER membrane. Okay, that is why the major function of the ER is lipid synthesis. So lipid synthesized in the ER, and the orientation, such a two leaf left will have been determined there, and they were budding out the vesicle, and they will kind of follow this orientation. And even when they fuse with the plasma membrane through could be their destination, they still follow this orientation. And even the protein orientation, this side and this side could be the N-terminal or C-terminal. They always follow this orientation because it's not easy for the lipid to do the flipping. Okay, so they follow this asymmetric. And it's also not easy for the member protein to do the flipping. They also need to kind of just like the lipid. They also will face the same issue. So they will follow this orientation without any other energy. Okay, and then until their destination. So the exoplasmic leaf left will contain more sphingomyelin and phosphatidylcholine. Okay, in the exoplasmic leaf left. And in the cytosolic leaf lab, will be more phosphatidyl ethanoamine, phosphatidyl and phosphatidyl in the cytosolic leaf lab. And the cholesterol, they distribute evenly in both leaf lab. Okay. So, but several molecules actually can regulate this asymmetry. Okay, it's kind of flippeds and scrambleds. Flippeds, this is this is a lipid bilayer. Okay, and this is the extracellular space, and this is the cytosol space. Okay, this is the extracellular space. Okay, so some flippeds, they can use energy from the ATP hydrolysis to move the lipid from the cytosolic leaf lab to the extraplasmic leaf lab. And that is why it flap out, okay? And the flip pass is also need energy, okay? It will flip the lipid in the exoplasmic leaf lab to the cytosolic leaf lab. They need energy, mainly through the ATP hydrolysis. And scrambleds, they don't need energy. Why can they kind of change such a lipid between the different leaf labs? Because they provide a hydrophilic road. It depends on their protein structure. They have a kind of hydrophobic interior property, so the lipid will be easier. They have a lower threshold to do the exchange. Okay, they don't need energy. So the scrambleds, they will break down the asymmetry if the cell need. Okay, but the flippeds and the floppeds, they need energy to do the specific work. Okay. So if cell want to disrupt such an asymmetric, they will start to express scrambleds and gradually to do this. Okay? If they want to build a specific asymmetry, they will use flippeds or floppeds, and they use energy to do this. Okay, and yeah.
Why they need to flip-flop movement?
Why? Because they need to accumulate certain their desired lipid to that leaf lab. For example, for the PS, which is the phosphatidylserine, they need to move the phosphatidylserine in the cytosolic leaf lab. Because if the phosphatidylserine move to the surface of the cell, it's kind of eat me signal. The macrophage will recognize any cell with surface phosphatidylserine and eat them, because those cells could be considered apoptotic cell. When cell trying to die to do such a programming cell death, they will kind of keep changing their lipid, and a lot of phosphatidylserine will move to the surface. Then there will be the eat me signal. So macrophage will start to see the phosphatidylserine and eat that cell. Okay, so you cannot do that. So it's such a lipid asymmetry is important to trigger many different things. So they need to be controlled. Okay, yeah. Okay, so this is just the table from the textbook. It's kind of boring. It just showed, they just purify the lipid from the different species and from different organelle, and use mass spectrum to kind of calculate the percentage of a PC, PE, PS, or sphingomyelin or cholesterol. So, of course, the first is lipid composition is different in different organelle. Yeah, okay. And the second, cholesterol doesn't exist in most of the prokaryotic cell. That is why you don't see that in the plant or E. coli. It will be the 0. Okay, you only can detect the cholesterol in the animal sample. And also, the ER and the Golgi are the main site to synthesize sphingolipid or P-phosphatidylcholine. So that is why you will see the higher concentration of those PC or PE or PS in the Golgi or ER here. Okay, that's it. Okay, and the different composition of the lipid can determine the thickness and the curvature of the membranes. For example, if the lipid two leaf lab, they are built up by the phosphatidylcholine, the thickness will be about 3.5 nanometers. But if they bind with the cholesterol will further extend their fatty acid tail. So it will kind of further extend the thickness to the 4 nanometer. Okay? But sphingomyelin will be the 4.6 to 5.6 nanometer and it's already pretty straight. So binding or crystal would not further extend their thickness. Okay, and PC is more column-shaped lipid, and the PE is a kind of cone shape of lipid. So it will also kind of accumulation with a cone shape or column shape will also change the curvature of the membrane. Okay. And also based on the mosaic fluid model, the mosaic turn is means some lipid they are not evenly distributed in this two-dimensional sheet. Sometimes they will kind of accumulate or form a cluster. So the one example is lipid raft. Okay, so this is a lipid raft. So it's not random distribute. The cholesterol sphingolipid and some specific protein will accumulate here. Okay, they call the lipid raft. The fluidity of the lipid raft is less than the surrounding area. That is why they don't kind of escape outside the lipid raft. They will accumulate here. Okay, and there are many. Usually, you will find many different signal pathway or receptor in the lipid raft. The cell will use this lipid raft to accumulate the receptor to the certain concentration. They need to trigger the cell signaling. The receptor need to above the concentration of the receptor need to above the certain threshold. If you don't have enough receptor on the surface locally, the ligand binding, for example, one ligand bind to the one receptor will not trigger anything. They don't accumulate enough downstream signaling, so they don't make any change. You need to accumulate maybe, if the threshold is 10, you need to accumulate 10 receptor locally and bind with the 10 ligands, then they will accumulate 10, which above the threshold of downstream effector, then they will make change. Okay, so this is the way lipid raft will be the way the cell to accumulate signaling receptor on the cell surface. There will be other ways called the lipid phase separation. Do you know that? Liquid-liquid phase separation. It's kind of cell can also form lipid droplet. It's not. I should not say the droplet, lipid droplet. It's just droplet. Okay, the protein will form the kind of droplet, and accumulate many different signaling molecule inside the cell. Okay, LLPS. You probably phase separation. Doesn't ring any bell? Phase separation. Oh, maybe it's phase transition. So cell sometimes they kind of, if the protein or signal molecule, different signal molecule, they have a different interaction, like this multi-tandem interaction with each other, they can form the kind of droplet inside the cell. Okay, and by doing that, they can increase the local concentration of those signal molecule compared to the other side of the cytosol, then trigger many different signaling. Yeah, but lipid raft will be the kind of the way they can accumulate these signaling molecules on the plasma membrane. So this lipid raft can be visualized because people already know certain signaling or certain molecule they will specifically retain here. For example, chlorotoxin B, which is a pathogen subunit B kind of molecules. People already know this pathogen subunit protein really they will go into the cell through the lipid raft. Okay, so you will see here they label in the red colors. And GN1 will be the glycosphingolipid, which will be the other marker of the lipid raft. You will see they overlap, colloquialize with each other. The purple will be the cell nucleus, and this is the cell. The boundary will be the cell's plasma membrane. Okay, and of course people can also use super resolution microscopy to visualize those lipid raft. So this will be the other marker. CTB will be the chlorotoxin B, which I showed you here. Okay, here they use this green color to label chlorotoxin B, and EGFR is a kind of receptor. Okay, you will see many EGFR concentrate in the lipid raft, and they are not evenly distributed on the cell surface, right? You can see many dots. Those are lipid raft on the cell surface. And also, I mentioned before, the ER will be the major site to synthesize the lipid. If ER synthesize too much lipids, they kind of will accumulate cholesterol in this the space between the two lipid raft. Okay, and finally they can build up such a micelle structure. Okay, this will be the lipid here. It could be the cholesterol, it could be the triglyceride. So those lipids are hydrophobic. They will align with those fatty acid tail, and also they build up this, we call this a lipid droplet. It's a kind of micelle. It's not the lipid bilayer. It's only one layer of the lipid. And the fatty acid tail will face to this lipid. Okay, and the lipid composition is similar to the cytosolic leaf lap of the ER because they build up budding out through the cytosolic leaf lap of the ER. Okay, you need to remember this. Okay, so as you can, and those are lipid droplets. If you eat too much Western food, I should not say McDonald's, Western food, okay, you will kind of accumulate many lipids. Inside in your kind of fat cell or adipocyte, you can kind of really see those lipid droplets. You can use a certain chemical dye to see those red color, those are lipid droplets, okay? And also, we can also use a live cell image to real-time monitor the formation of those lipid droplets, as you can see here. After you incubate cell with certain fluorescent lipid, you will start to see more red dot here. Those are network, they are ER. And you will start to see many red dot here. Those are lipid droplets. They form from the ER, okay? Okay, so just give you the overview. First, we talk about this on how those molecules behave through the fluid mosaic models. And the second, we talk about this will be the orientation will be the hydrophilic polar head and the hydrophilic polar head, hydrophobic layer, and the hydrophilic polar head, the other sides. And those are phospholipids, they are amphipathic. And also we talk about the three different major lipid phosphoglycerides, sphingolipid and steroid. And also we was claimed that the membrane is asymmetric. There will be more PCSM in the exoplasmic leaf lap, and more PE, PS, and PI in the cytosolic leaf lap. And the cholesterol will be the evenly distributed to the both sides. So that is why the PS, in the normal condition, they will accumulate in this side. If in the apoptotic cell, they flip to the other side, then they will be kind of become an eat me signal. Okay. And lipid composition will change their thickness and the shape of the membrane, and lipid protein can laterally diffuse within the membrane, and their dynamic can be measured by FRAP. The lipid raft, it could be the cholesterol, sphingolipid, and a certain protein-rich microdomain. Okay, and I want to mention this about the FRAP. So FRAP has been widely used to measure the membrane molecules. Can FRAP be used to measure the soluble molecules dynamic? What do you think? It's only kind of, we just showed you to always talk about the membrane molecule, right? But there's a lot of things moving in the cytosol, right? Can FRAP be used to measure the dynamic of soluble molecules? Yes or no? The answer is yes, but the soluble molecule, they move too fast, super fast, okay? So the bleaching to the region of interest, kind of certain local area, bleach that area, it takes about the 10 seconds. But within the 10 seconds, every molecule can go around the whole cell. So you just keep bleaching the one area, they recover during the photobleaching. Okay, you never see the bleaching. If you keep bleaching, you don't, why I cannot see the bleaching? Do those lasers still functional? You just keep bleaching, and then finally, the whole cell becomes dim because you just keep bleaching the molecule, go into that area. And finally, the whole fluorescence molecule in the cytosol, they're gone. Okay, so it's just technically difficult to measure the dynamic of the soluble molecule because they go too fast. Every 10 seconds. For example, the lateral diffusion, theoretical speed is one lipid can theoretically move from the 1 micrometer per second, but for the soluble one, it could be the 100-fold faster or 1,000-fold faster. Yeah, you have no chance to kind of use a FRAP to measure that. Okay, so let us take a 5-minute break. So.